Have you ever wondered how chemists detect substances, determine their concentration, or even understand their behavior? The answer lies in electroanalytical techniques. Welcome to the fascinating world of chemistry, where we unravel the mysteries of matter through various analytical techniques. One such technique is electroanalytical chemistry. This is a subdiscipline of analytical chemistry that employs electricity and chemistry in tandem to derive meaningful data about a substance or a reaction. Now you might ask, why is this important? Well, electroanalytical techniques offer a unique way to probe chemical phenomena. They allow us to detect minute amounts of substances, determine their concentration, and even glean insights into their behavior. This is vital in a multitude of fields, from environmental science to pharmaceuticals, forensic science to food quality control, and beyond. So, what exactly are these electroanalytical techniques? Essentially, they are a set of methods that use electricity as a tool to study chemical compounds and reactions. This could be anything from measuring the voltage across a solution to determine its pH, to applying an electric current to a sample to induce a chemical reaction. These techniques hinge on the principle that every chemical compound possesses unique electrochemical properties. By studying these properties, we can identify and quantify the compound, and even infer its structure. This is achieved through the careful manipulation and measurement of electrical signals, such as current, voltage, or charge, which are related to the chemical properties of the sample under investigation. But how do we manipulate and measure these electrical signals? This is where the magic of electroanalytical techniques comes into play. They use a range of instruments and methods, from electrodes to potentiostats, voltammetry to spectroscopy, to generate, measure, and analyze these signals. Now that we have a basic understanding of electroanalytical techniques, it's time to delve deeper into the types of these techniques. So stay tuned as we embark on this exciting journey into the electrifying world of electroanalytical chemistry. Electroanalytical techniques are not limited to one type. In fact, they come in a variety of forms, each with unique characteristics and applications. First on our list is potentiometry. This technique is all about measuring the potential of a solution without drawing any significant current. A common example of this is the pH meter, which is widely used to determine the pH level of various solutions. The beauty of potentiometry is its simplicity and directness, making it a popular choice in many practical applications. Next up, we have voltammetry. Now, this one is a bit more complex. Voltammetry is about controlling the potential of an electrode and measuring the resulting current. It's like asking a question to an electrode and waiting for its response in the form of current. It's a key player in the world of analytical chemistry, with applications ranging from testing water quality to detecting metal ions. Finally, let's talk about coulometry. This technique is based on the measurement of electrical charge. In coulometry, we let a chemical reaction proceed to completion, and then we measure the total quantity of electricity required for the reaction. This allows us to determine the amount of a substance present in the sample. Coulometry is particularly useful when dealing with small amounts of substances, thanks to its high accuracy and sensitivity. So, you see, these types of electroanalytical techniques have their unique features and applications. Potentiometry gives us direct and straightforward measurements. Voltammetry allows us to probe the behavior of electrodes, and coulometry provides us with a precise way to measure substances. They all play crucial roles in the field of analytical chemistry, each with its own set of advantages. Knowing about these types of techniques gives you a broader understanding of electroanalytical techniques. Now, let's discuss the electrochemical cell. The heart of most electroanalytical techniques is the electrochemical cell. But what is it, and how does it work? An electrochemical cell is a device that can generate electrical energy from a chemical reaction, or use electrical energy to cause a chemical change. Imagine two containers, each filled with a solution of a different substance. In each container, we place an electrode, which is a conductor through which electricity enters or leaves our cell. The magic begins when we connect these electrodes with a wire. This initiates a chemical reaction, causing electrons to flow from one electrode to another. The electrode that loses electrons is called the anode, while the one that gains electrons is the cathode. And voila, we have an electric current. Now you might wonder what drives this electron transfer. This is where the concept of potentials in an electrochemical cell comes into play. The potential difference or voltage between the two electrodes is what pushes the electrons to move. The greater the potential difference, the greater the force driving the electrons, and thus, the stronger the current. But how do we calculate this potential difference? Enter the Nernst equation. Named after the German scientist Walter Nernst, this equation allows us to calculate the voltage of an electrochemical cell. It takes into account the concentrations of the substances involved, the temperature, and the number of electrons transferred in the reaction. In essence, the Nernst equation helps us understand that the potential of an electrochemical cell is not a fixed value, but rather, it can change based on the conditions under which the cell operates. So, to wrap it up, an electrochemical cell is a neat little device that allows us to convert between chemical and electrical energy. The potential difference between its electrodes drives the flow of electrons, and the Nernst equation helps us calculate this potential. With the knowledge of electrochemical cell and potentials, we can further explore the current-potential relationships. The current and potential relationship is a fundamental concept in electroanalytical techniques. But how do they correlate? You see, in an electrochemical reaction, current is the rate of flow of charge, while potential is the driving force that propels this charge. The relationship between these two is governed by the Nernst equation, which provides a quantitative measure of this relationship. Now let's touch upon the concept of mass transfer. It's a process that describes how mass moves from one location to another, and it plays a pivotal role in electroanalytical techniques. Mass transfer occurs by three mechanisms: migration, convection, and diffusion. Migration refers to the movement of ions under the influence of an electric field. This movement is driven by the potential difference across the cell. Next, we have convection. Convection is the movement of molecules within fluids, such as liquids or gases, and is caused by temperature differences within the fluid. In electroanalytical techniques, convection can occur naturally, or it can be forced by stirring or shaking the solution. Lastly, we have diffusion. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. It's a passive process, driven by the need to achieve equilibrium. In an electrochemical cell, diffusion is a key process that drives the movement of ions. Together, migration, convection, and diffusion work in harmony to facilitate mass transfer in an electrochemical system. They ensure the uniform distribution of ions, which is crucial for the functioning of the electrochemical cell. Understanding these concepts is crucial to mastering electroanalytical techniques. But remember, practice makes perfect. Let's take a moment to revise what we've learned about electroanalytical techniques. We kicked things off by introducing electroanalytical techniques, a crucial subset of analytical chemistry that uses electrical and electrochemical phenomena to measure chemical quantities. These techniques can tell us a great deal about the composition and concentration of a chemical sample, making them indispensable in laboratories worldwide. Next, we explored the different types of electroanalytical techniques. We learned about potentiometric methods, where no current flows, and voltammetric methods, where current is the measured variable. We also touched on coulometric methods, where the quantity of matter is determined by measuring the quantity of electricity in coulombs. Each of these techniques has its own unique applications and advantages. Then, we delved into the heart of these techniques, the electrochemical cell. We learned how it's a system that can either generate electrical energy from chemical reactions or use electrical energy to cause chemical reactions. We also discussed potentials in electroanalytical cells and how they can be measured using the Nernst equation. This equation beautifully links the voltage, temperature, reaction quotient, and number of electrons transferred in a redox reaction. Moving on, we explored the fascinating relationship between current and potential. We discovered how the flow of current can be influenced by the potential difference applied, and how this can be mapped out using current potential graphs. This relationship is pivotal in understanding the behavior of electrochemical systems. Lastly, we discussed mass transfer by migration, convection, and diffusion. These processes occur in an electrolyte solution during a reaction, and understanding them can provide valuable insights into the behavior of the system. With this knowledge, you are now equipped to delve deeper into the world of electroanalytical techniques. Remember, the world of chemistry is vast and constantly evolving. Subscribe for more because you won't find me later.